Hello, and welcome to the Chapter 12, Part 3 lecture on cell division. You should use the information in this lecture to complete the Chapter 12, Part 3 guided notes, which of course you should complete before you attend class. In Parts 1 and 2 of Chapter 12, we discussed a lot of preliminary information about the cell cycle and chromosomes and chromatids that you will now need to apply in learning about division. Reproduction is sometimes called the biological imperative, and that's because this is something that all living things have to be able to do in order for the species to remain on the planet. There are many different ways to reproduce. In asexual reproduction, the organism will make an exact copy of itself. So in other words, it will clone itself, and the offspring that are produced will be genetically identical to the parent that produced them. This is useful for making more individuals, like when bacteria go through asexual reproduction, they make more bacteria. But it's also useful in multicellular organisms for repair. So for instance, when your body is healing a wound and the cells are dividing, those cells will replace the damaged cells. That's a form of asexual reproduction too. Here we see several different examples of asexual reproduction. Here we have simple mitosis, or cell division, and this cell is dividing into two new individuals. Both of these individuals are genetically identical to one another. Here we see some yeast cells performing something called budding. Budding is another form of asexual reproduction, wherein the cells kind of hang on to one another. They don't separate completely, so they make this clump of cells. But all of the yeast cells within this clump, or this bud, are genetically identical to one another. Multicellular organisms can sometimes use asexual reproduction to make new individuals as well. Humans don't do this, but lots of invertebrates do. Here, for instance, we have a little critter called a hydra. This is a little invertebrate organism. It's microscopic, and it lives in fresh water sources. It's related to a sea anemone. And what it's doing here is asexual reproduction. This is also referred to as a form of budding. And so it's performing cell division and growing a little baby on its side. So this little thing that looks like a waving arm is actually a little baby hydra. Eventually this little baby hydra will break off of the parent and fall off and start to grow on its own, and it will be a clone of the parent. So there's many different forms of asexual reproduction that exist. Prokaryotic cells often use a type of division known as binary fission. Binary fission is far simpler than mitosis. This is because prokaryotic cells have a lot less DNA than eukaryotic cells, and the machinery needed to move that DNA around is also a lot simpler and less complex. Prokaryotic DNA is stored in a single chromosome known as the bacterial chromosome, and it's held in a little loop inside the cell. In order to divide, all that cell has to do is make a copy of that loop, glue the two copies down, and then divide the cytoplasm in the cell wall in between the two copies to make two daughter cells that are identical to one another. It's simple, it's fast, and it's one of the big reasons that bacteria can reproduce so much faster than our own cells. Cell division in eukaryotic cells is, of course, much more complicated than in prokaryotic cells. One big reason for this is the amount of DNA found in these different cell types. In prokaryotic cells, there's just a single chromosome that's connected together in a loop. And so the prokaryotic cell has that loop and a copy of the loop to keep track of, and that's it. Inside eukaryotic cells, there are many chromosomes. Inside a human cell, for instance, there are 46 chromosomes, and those get copied before division begins. So there's actually 92 of those pieces of DNA floating around that need to be moved in a particular pattern in order for division to be successful. So there's a lot more machinery associated with eukaryotic division than prokaryotic as well. All that machinery helps move the DNA around. These machines include things like spindle apparatus, microtubules, and motor proteins, and we'll see them in action in a little bit. Two major types of division exist in the eukaryotic system: mitosis and meiosis. Mitosis represents that asexual reproduction that we were talking about before. So that's the process by which cells are trying to make exact copies of existing cells. Again, this process is handy for growing a body and also for healing it. So for instance, when cells deep inside your skin in the dermis reproduce, they can replace the skin cells coming off of the top of the epidermis that we flake off all the time. Meiosis is more involved in making sex cells, things such as egg cells and sperm cells, of course. And so it's more involved in sexual reproduction. We're going to discuss it in more detail in chapter 13. Cell division actually involves two separate processes that happen simultaneously. The division of the nucleus is mitosis or meiosis, so that's the splitting up of the DNA. The splitting up of the rest of the cell, the cytoplasm and the organelles, is known as cytokinesis, which literally means cell movement. Before we get into all those fun phases of mitosis and all that neato machinery that makes it happen, let's just take a moment and discuss the point of mitosis. Remember that the point of mitosis is to separate the DNA accurately. So what the cell will do is it will first go through a process of DNA replication. And if you remember, this happens in the S phase of the cell cycle. So during the S phase, it will make a complete copy of its DNA. And then in the M phase, the mitotic phase, or the division phase, what the cell is going to need to do is to split up those copies so that each resulting daughter cell has an exact copy of that DNA. It has a complete set of instructions for being that type of cell. Splitting up the DNA into two identical sets is a really, really important job. If the cell makes a mistake, which can happen, and the resulting daughter cell is missing a piece of DNA, it's going to have problems. If the cell makes a mistake and a daughter cell ends up with an extra copy of a piece of DNA, it's probably also going to have problems, although different problems than if it were missing some DNA. So think of it this way. If we have a parent cell that is diploid with a chromosome number 46, as we have in human beings, by the end of mitosis, what we want to end up with are two daughter cells, and we want those daughter cells to be identical. So we want each of the daughter cells to be diploid, and we want each of the daughter cells to have 46 chromosomes. Mitosis can occur in haploid cells as well, not in the human system, but in things like fungus. Sometimes you see this happening. But if the cell starts out haploid, the daughter cells are also going to be haploid. So the point of this is that the chromosome number is going to stay the same from the parent cell to the daughter cells. They're genetically identical to each other. So the chromosome number in mitosis stays the same from the parent cell to the two daughter cells. Really, the only thing that changes through mitosis is the number of cells. We start out with one parent cell and we end up with two daughter cells. How can this be physically achieved? Well, mitosis is really a process of separating chromatids from one another. Let me show you what I mean. Here we have a simplified cell. Instead of having a 2n diploid number 46, it just has a diploid number of 2. You can tell that this cell is diploid because it has two copies of this chromosome, one that came from mom and one that came from dad. In the S phase of the cell cycle, those chromosomes are going to get copied. So if we could see the chromosomes after the S phase, they might look like this. They look like X's now. During the mitotic process, each of these duplicated chromosomes is going to get split in half. So each chromatid within that chromosome is going to go separate directions to opposite ends of this cell. They're going to separate into two equal sets. And so now you can see that at the end of this process, these two daughter cells are identical to one another, and they are identical to the parent cell that we started with. They have the same number of chromosomes. Those chromosomes carry exactly the same information. So everything stayed the same except the number of cells. A note about the use of the words chromosome and chromatids. In this picture, we have two chromosomes. They are unduplicated, so they look like little bars. After the S phase, they become duplicated chromosomes. So now they look like X's, and just to remind you, each contains two chromatids, sister chromatids. As they travel together, these chromatids actually comprise one chromosome. So that's a chromosome right there. But the instant they separate from one another, which happens later in mitosis, these chromatids now become chromosomes. So in this picture, we actually have two duplicated chromosomes, but in this picture, we actually have four unduplicated chromosomes because they've split up from one another. They're no longer called chromatids. So now that you understand the overall process of mitosis and how chromatids are separated from one another, we can move on and talk about the stages of mitosis and the machinery that actually moves this DNA around. There are 5 important stages to mitosis. Now, if you've studied biology in the past, you may be thinking, hey, aren't there only 4 stages of mitosis? Why are we talking about 5 things? Well, prophase was recently split into 2 smaller phases. So now we have prophase, prometaphase, which used to be called late prophase, metaphase, anaphase, and telophase. A handy memory trick for this is PPMAT, or please, people must ace test. Or if you prefer, you can always just remember that it makes a word, PPMAT, which is what you put down if you have a new puppy. The very first phase of mitosis is known as prophase. Remember that pro means before or beginning. At this phase, the cell is going to get ready to divide. So the DNA, which up to now has been in its loosely wound chromatin form, is going to begin to wind up into visible chromosomes. Now those chromosomes have already been duplicated, so they will look like X's with their little chromatids and their little centromere in the middle. A couple of other important pieces of machinery are also going to wake up and activate. First, we have these things called centrioles. You may remember centrioles from way back when we talked about organelles. Centrioles are like little bundles of branches, and these little bundles of branches, pieces of cytoskeleton, are going to begin to grow, and they're going to send out these long tubes. The tubes are what are going to be used to move the DNA around, and they are made of microtubules. Eventually, these tubes are going to form a big structure called the mitotic spindle. The second phase of mitosis, which we used to just call late prophase, is now referred to as prometaphase. Several important things are going to happen during this phase. For one thing, we're going to get rid of the nucleus. The nucleus has that big, thick double membrane that's surrounding all that DNA, so we need to get rid of it. It's going to disintegrate and fall apart so we can move the DNA around. The second important thing that's going to happen is that those spindle fibers that have been growing are now going to reach out and they're going to connect to the coiled up chromosomes. So it looks a little something like this. Here's your chromosome, the little centromere in the middle. Each spindle fiber is going to reach over and it's going to grab onto either side of the chromosome on each side at an area called a kinetochore. These ropes are made of cytoskeleton. So remember that cytoskeleton can grow and shrink. So these pieces of rope are going to start pulling and pushing on the DNA, and it's going to start moving it around in a particular pattern. The third phase of mitosis is called metaphase. At this point, the centrioles reach the opposite poles, and those protein ropes, those spindle fibers that have been pushing and pulling on the DNA, are going to get it all lined up right in the middle of the cell. They line up the DNA, the chromosomes, along an imaginary line called a metaphasic plate. and notice how they line them up. Here we have 2 chromosomes, for instance. The cell's going to line those chromosomes up so that this imaginary line runs right down the middle between the 2 sister chromatids. In anaphase, the 4th phase of mitosis, the sister chromatids are going to separate from one another. This action is accomplished by the centrioles and the spindle fibers. The spindle fibers during anaphase are going to shorten. So they're going to get smaller, and that's actually going to pull on those chromatids and break the chromosomes apart. And it's going to move those chromatids towards opposite poles. At the same time, there are spindle fibers that have formed between the 2 chromatids, and those spindle fibers are actually going to push on the chromatids. So there's both a simultaneous pushing and pulling of the DNA that makes the chromatids separate and drags these chromatids away from each other towards opposite poles. Once the 2 sets of chromosomes have reached opposite poles or opposite ends of that cell, the 5th phase of mitosis, known as telophase, can begin. Telophase is really the beginning phases of mitosis in reverse. So think about what happened in prophase and prometaphase. The DNA coiled up into chromosomes. Well, in telophase, that's going to reverse. The chromosomes are going to uncoil a little bit back into that chromatin form. Also, in prometaphase, the nuclear envelope disappeared. So in telophase, we have to build 2 new nuclear envelopes to surround those 2 new sets of DNA. Also in the beginning phases of mitosis, the centrioles begin to activate and make spindle fibers. So at this point, the centrioles are going to deactivate and the spindle fibers are going to break down because they're no longer needed. Notice that cytokinesis is also beginning during telophase. It's technically a separate process, but it kind of starts happening at the same time. The division of the cytoplasm in organelles is known as cytokinesis. It begins at about the same time as telophase of mitosis. Cytokinesis can be accomplished a couple of different ways depending on the type of cell you're talking about. In animal cells, they're actually little pieces of the cytoskeleton that will pull together, and they grow these strings that pinch the cell in two. In cells that have a cell wall, however, cytokinesis is more complicated. They actually have to grow pieces of wall in between the 2 cells, and the growth of these pieces of wall actually kind of pull the membrane in two and separate the 2 sets of cells. So it's more complicated in plant and fungal cells.
Cell division is required for an organism to grow, mature, and maintain tissues. During the mitotic phase, a cell will undergo mitosis to form 2 new nuclei, and then divide to form 2 new individual cells during cytokinesis. Mitosis is the process of dividing the duplicated DNA of a cell into 2 new nuclei. Mitosis is split into distinct stages. The first stage is prophase. The DNA condenses, organizes, and the classic chromosome structure appears. Next comes prometaphase, where microtubules attach to the chromosomes. This step is followed by metaphase, where the chromosomes align. Metaphase is followed by anaphase, where the chromosomes separate. Finally, during telophase, nuclear membranes reappear around the 2 sets of chromosomes. Mitosis is now complete. After mitosis, 2 new cells are formed by a process called cytokinesis. Mitosis is only one part of what is called the cell cycle. For many eukaryotic cells, a cell is duplicated every 24 hours. Most of the life of a cell is spent in interphase. Interphase consists of 3 stages called G1, S and G2. G1, or gap 1, is the first growth stage of interphase. In G1, the cell grows to nearly its full size and performs many of its specific biochemical functions that aid the organism. Next is the S, or synthesis phase. This is an important stage because it is during the S phase that DNA in the nucleus is replicated. The cell next enters another growth stage called G2, or gap 2. It is during G2 that the cell finishes growing. Once the cell has duplicated DNA in the nucleus and 2 centrosomes have appeared in the cytoplasm, mitosis can begin. For a typical eukaryotic cell, this will last about 80 minutes. During the first stage of mitosis, called prophase, we first see the classic chromosome structure. This occurs through a condensation process. At the same time, protein strands called microtubules appear from the centrosomes in animals. Finally, a structure found within the nucleus, the nucleolus, disappears. Next, prometaphase begins when the nuclear membrane is broken down. At the same time, microtubule strands, or spindle fibers, are growing from the centrosomes. These strands attach to a protein structure called the kinetochore. One kinetochore is attached to the centromere of each sister chromatid. Next comes metaphase. During this stage, the sister chromatids align along the center of the cell so that both chromatids face toward opposite poles of the cell. Now the sister chromatids are ready to be separated. This occurs during anaphase through a shortening of the microtubules attached to the kinetochores. Additionally, the poles of the cell move farther apart, causing increased separation of sister chromatids. At the end of anaphase, the sister chromatids have moved to the 2 ends of the cell. Telophase is the final stage of mitosis. It is here the components of the new cells begin to appear. At this point, the spindle fibers are broken up. A new nuclear membrane surrounds the chromosomes at the end of each cell, and the chromosomes uncoil and return to an uncondensed state. Mitosis is now complete. The formation of 2 cells is all that remains. Following mitosis, the cell undergoes a process called cytokinesis. First, the cell is compressed by a contractile ring that divides the cell in nearly equal halves. By now, the organelles in the cell have been replicated and are now divided between the 2 halves of the cell. This includes mitochondria, Golgi bodies, and the rough ER. Plant cells also have chloroplasts. Once split, the 2 new cells are now fully in the G1 stage of interphase and ready again to begin their growth. Let's watch the process one more time. Mitosis begins with prophase. Notice the DNA condensing into chromosomes during this stage. Microtubules appear during prometaphase, and the nuclear membrane breaks down. Metaphase occurs when the chromosomes are aligned at the center of the cell. During anaphase, the chromosomes are moving apart. The telophase stage is marked by the appearance of new nuclear membranes. This is the end of mitosis. Finally, the splitting of the cell occurs during cytokinesis. The 2 new cells are now ready to grow and perform their specialized functions.
All right, you guys, you did it. You completed Chapter 12 and you now have all the basic information you need about mitosis. Make sure you complete your notes in those year turns and bring them to class because we're going to work with this process some more.